Warm would be calar, calor. Pain would be dolar. Another type of cell found within connective tissue is the plasma cell. Plasma cells originate from B lymphocytes, and their function is to produce antibodies. So they're responsible for production of antibodies. You see other types of white blood cells besides lymphocytes. You may see, for instance, neutrophils. How do they get there? Well, they leave the bloodstream, pass through the blood vessels into the tissue by a process called diapedesis. Diapedesis refers to the process where a cell squeezes through the lining, the endothelial lining of the blood vessels to enter the tissue. If tissue is inflamed, white blood cells are going to move into the tissue. So that's one of the characteristics of inflammation. When you look at it under the microscope, you see lots of white blood cells. And then finally, you may see fat cells. Sometimes these are called adipocytes. These are cells that simply house triglycerides or fat. Now, earlier in our first talk, we said that connective tissue originates from mesoderm. Okay. Mesoderm will differentiate into embryonic connective tissue, which includes mesenchymal cells. And those mesenchymal cells, or these are types of cells present in mesenchyme, can then differentiate along multiple pathways. These are a type of stem cell. They can produce fibroblasts, adipocytes, chondrogenic cells, the types of cells that are present within cartilage, or osteoprogenitor cells, which are the types of cells that will be present within bone. Mesoderm also leads to formation of other blood cells, lymphocytes, other white blood cells, platelets, red blood cells. Let's look at these connective tissue cells. On the left here, these are fibroblasts. Fibroblasts are long, spindle-shaped cells with flattened nuclei, which are typically dark staining. If you have a really good stain, you can see some of the cytoplasm extending beyond the nucleus. Many times it's difficult. So all you see is that nucleus. Okay? And remember, the function of these cells is to synthesize the surrounding extracellular matrix. On the right-hand side, these cells that are filled with granules, those are mast cells. Those are the types of cells that are active in inflammatory responses. If you think about your nasal mucosa, in other words, the mucous membrane that surrounds your nasal cavity, okay, like all other types of connective tissue, that mucous membrane houses mast cells. Let's suppose this is a mast cell. If you're allergic to pollen or ragweed, okay, you're going to have IgE antibodies that are tethered to the surface of these mast cells. These are antibodies to pollen or ragweed, whatever you're sensitive to. And so during spring, when everything's in bloom, if you breathe in some ragweed, it will bind to these IgE antibodies and trigger degranulation of the mast cells, releasing histamine and other inflammatory mediators. So what happens? Your nasal mucosa becomes congested, swollen, it becomes red, okay, perhaps itchy. Okay, those are all characteristics of inflammation. These are plasma cells. Plasma cells are interesting. They're typically oval shaped, although they can be a little bit more rounder at times. Like this, and the nucleus is skewed to one side. The chromatin in the nucleus is oftentimes clumped. So it may even resemble like a clock face. We think of these clumps as being like numbers that's around the base of a clock. Okay? Then you're going to have a little pale area adjacent to the nucleus, and then a darker staining region at the opposite end of the cell. Okay? So this more basophilic or darker staining region, which appears somewhat granular, those are ribosomes. Remember, plasma cells are active in the production of immunoglobulins or antibodies. So ribosomes are the site where proteins are synthesized. So you're going to have a lot of ribosomes. This clear area next to the nucleus is the site of the Golgi apparatus, where these antibodies are packaged before being excreted by the cell. Okay? So this is kind of the architecture of a plasma cell. So here is the nucleus, it's skewed to one side. Here's that clear zone. Some people call this a HOF. And then here's the more basophilic region at the opposite end, where the immunoglobulins are synthesized. Okay. We'll take a look at adipocytes later when we talk about fat. But I think you're familiar with those. Let's talk about the types of fiber present within the extracellular matrix. There are three principal forms of fibers, or types of fibers. One is collagen, the second is elastic fibers, and the third is reticular fibers. Collagen fibers are probably the most common. Now the truth is, there are a good number of different types of collagen fibers. If we don't specify otherwise, we're talking about type 1 collagen. Type 1 collagen is the strongest type of collagen fiber. Okay? These are the most numerous fibers in connective tissue. These fibers provide for tensile strength and resistance to tension. Okay? These are called white fibers because they look white. They have a glistening white appearance in living tissue. Okay? So, for example, you see collagen fibers in tendons and ligaments. So if someone's undergoing an operation on their knee, okay, and you open up the knee, you'll see that the patella ligament has a glistening white appearance because it's made largely of collagen fibers. And that makes sense because the ligament is connecting bone to bone. It's got to be strong. Elastic fibers are formed primarily from the protein elastin. These fibers tend to be like rubber bands. They can stretch, okay, like a rubber band and recoil. In living tissue, elastic fibers, again, this is in living tissue, they have a yellowish gleam. So sometimes surgeons will call these yellow fibers. Then the third type of fiber is called reticular fibers. And reticular fibers are really type 3 collagen, and we'll talk about collagen in a little bit. But type 3 collagen is a lot thinner. It tends to branch. Okay? It's not as strong in terms of tensile strength. Okay? So these type 3 collagen fibers are referred to as reticular fibers. The other thing is because they're thin, they don't show up using routine stains. In other words, they simply don't pick up enough H&E, methoxylin and eosin, so that they're visible by light microscopy. If you really want to see reticular fibers well, you have to use what's called a silver stain, because these fibers are agar-philic, meaning they're silver-loving. Now, typically, silver stains are stains that, when they're picked up by structures of fibers, those structures of fibers appear dark. Sometimes black, sometimes a vivid blue or purple. So you'll recognize right away when a different stain other than H&E is being used. And oftentimes we'll use these stains to demonstrate things like reticular fibers or reticular connective tissue. In AMP1, we talked about proteins in the very beginning. We said there are two types of proteins, structural proteins and functional proteins. Structural proteins, like collagen, tend to be linear molecules. Now, all proteins have a primary structure. The primary structure is simply the sequence of amino acids that make up that molecule. But structural proteins like collagen also have a secondary structure, meaning the molecules here have an alpha helical configuration, they form a helix. Okay? That's a secondary structure. And in fact, collagen has a quaternary structure because the molecule is formed of 3 different polypeptide strands that are linked together. That's referred to as a quaternary structure. Okay? So each collagen molecule is formed of 3 polypeptide chains with these alpha helical configurations. So we can call these alpha chains. And they're linked together to form a triple helix. Now, the truth is a collagen fiber is made of buku collagen molecules that are bundled together. Okay? Each alpha chain, 1, 2, 3, each of them, may have been produced by any of 42 different genes. Okay? And of course, these genes, what they do is they're translated into the alpha chain, and depending upon the gene, it may have a different primary structure sequence of amino acids. So because of the fact that these collagen molecules may be produced by up to 42 different genes, individuals can have different types of collagen fibers. There are different types of collagen fibers that exist in different tissues. In fact, there are 29 known alpha chain combinations. So that means there are 29 different types of collagen. Our main focus is going to be on type 1 collagen and reticular fibers, type 3. We said that the most abundant type of collagen is type 1. And it's got a high tensile strength. It's not very elastic. It doesn't stretch much. We said that type 3 collagen fibers are much thinner and branched. These are reticular fibers. Reticular fibers don't form bundles to the extent that type 1 fibers do. Okay, so that's why they're thinner, they're not as strong. So let's talk about how type 1 collagen is produced. Well, first, you have to produce the alpha chains. Okay, and of course, this synthesis occurs at the site of the ribosome. The process is called translation. In other words, when you take messenger RNA, and from that messenger RNA, you synthesize proteins, and that occurs at the ribosome, okay, you produce polypeptides, okay, and that process is translation. Once the polypeptides are produced, the alpha chains are produced, they self-assemble. They link up and form what's called a pro-collagen molecule. Now, this pro-collagen molecule undergoes some post-translational events, one of which is the ends of the molecule are clipped by pro-collagen peptidase. That results in a different molecule, slightly different, modified, smaller, called tropocollagen. Tropocollagen molecules will self-assemble, will bundle up to form fibrils, which in turn bundle up with other fibrils to form collagen fibers. Okay? So you have the initial translation, and you have a number of post-translational events. This all occurs within or outside of the fibroblast. In fact, think of this as a big fibroblast cell here. Here's the nucleus. Okay? And within the nucleus, you're forming messenger RNA. That's called transcription, when you form messenger RNA from the DNA template. That messenger RNA leaves the nucleus and docks on ribosomes within rough endoplasmic reticulum. Okay? On the ribosome, that messenger RNA gets translated into those alpha chains. Okay? Remember, you have 3 of those alpha chains. Those alpha chains then get hydroxylated. This is a post-translational event. Hydroxylation is dependent upon vitamin C. The next post-translational event is that these hydroxylated alpha chains then interact with sugars, like lactose or glucose. In other words, the chains get glycosylated. It means just little chains of sugars are added to the hydroxyl groups. Okay? Then these glycosylated alpha chains self-assemble, forming procollagen. Okay. Now, this procollagen, these procollagen molecules then enter into the Golgi apparatus. And in the Golgi apparatus, a group of procollagen molecules are packaged, meaning they're surrounded by a membrane-lined vesicle, and then they're excreted from the cell. Outside of the cell, the procollagen molecules get cleaved. Remember we said procollagen peptidase breaks off some of the amino acids at each end. And that forms what really is tropocollagen, what we referred to before as tropocollagen. And we said the tropocollagen is then going to self-assemble into fibrils, which in turn self-assemble into fibers. So the production of collagen involves numerous steps. We said that collagen synthesis is vitamin C dependent. If you have a vitamin C deficiency, that condition is called scurvy. If you have scurvy, you can't produce intact, functional collagen fibers. Okay, because remember this first post-translational event, hydroxylation is vitamin C dependent. So as a result, you're going to have defective collagen synthesis if you have scurvy. Your tissues are going to tear more easily. You're going to bleed more easily. Okay? So it can result in hemorrhages in the skin and the gums and poor wound healing. This was a problem faced by early explorers. They'd come across from the old world to the new world, and they'd be at sea for weeks or months. Okay? The fruit that they brought with them that contained vitamin C, that would rot within a week or two. And so they would develop vitamin C deficiencies if they didn't reach the new land quickly. Now, we said that there are multiple genes involved in collagen synthesis. Okay? And there are multiple post-translational events. So here it says collagen synthesis depends on the expression of several genes and several post-translational events. There are a number of clinical disorders that result from defects along this way, along this pathway. Again, this is just a picture of the different events that occur. Hydroxylation, where you put hydroxyl groups, attach those to the amino acids within the alpha chain. Then glycosylation, this is where you add sugars to some of these hydroxyl groups. Okay? If there's a defect with glycosylation, you can have weak collagen that's defective. And this can lead to a problem called osteogenesis imperfecta, which is called brittle bone syndrome. These are children whose bones break very easily. In fact, oftentimes the parents, before the disease is discovered, the parents are accused of child abuse because the kid goes into the ER frequently with a broken bone. If there's a defect further down, okay, with peptide cleavage, you can develop a disease called Ehlers-Danlos syndrome, where the joints are hyperflexible. So there are a number of disorders that relate to defective collagen synthesis. If someone has brittle bone disease, what happens is the bones don't have the strength that they typically do, so they tend to bow and break. Okay? So the brittle bone disease is caused by an abnormal gene for collagen type 1 molecules. The mutant gene codes for collagen alpha chains with abnormal structure. So therefore they form abnormal procollagen, which has an adverse effect on mineralization of bone and bone strength. There was a movie about someone with brittle bone disease. Did y'all see it? Unbreakable?
Oh yeah.
It was by M. Night Shyamalan.
It's the series.
Now, people with Ehlers-Danlos syndrome have hyperextensible joints. They can take their fingers and touch the backside of their wrist. Again, because of defective collagen. So there are a number of disorders that are marked by defective collagen synthesis. Let's talk about elastic fibers. The main component of elastic fibers is the molecule elastin. And these elastin molecules are linked together in such a way that these fibers can stretch and recoil. Okay? In fact, these elastin molecules can form fibers, and in some cases they form sheets or lamina. Another name for an elastic sheet is a lamina with the E, it's plural. Sorry. So tissues may contain elastic fibers as well as elastic sheets or lamina, made up of these elastic molecules that are linked together. Now, with regard to elastic fibers, if the blue here represents bundles of elastin molecules, these elastic fibers also contain a linear protein called fibrillin. And the fibrillin helps the structure maintain a more linear architecture. Okay? If you're looking at tissue, elastic fibers tend to be more refractile, stand out, they reflect light. Okay? So in this H&E tissue, the elastic fibers stand out as glass, bright pink stained structures. It's these. Okay? They have greater affinity, a greater attraction to eosin, which is the pink part of the H&E stain, than do other collagen fibers. Now, if you want to really identify elastic fibers, you can do two things. You can move the diaphragm, open it or close it. In one direction, tissue becomes more refractile. You can also move this condenser up or down. The condenser, let's see. Let's see what we move the condenser. It's just a knob here. For my